This is lecture 11 for Biology 178, covering cardiomyocyte physiology as well as the electrical system of the heart and electrophysiology. So as we finished with the previous lecture, we talked about cardiomyocytes, which are the contractile cells of the heart. These cells share characteristics with both skeletal and smooth muscle cells. Remember that cardiomyocytes, though, are found only in the heart. They are smaller than skeletal muscles, and they have a single centrally located nucleus. They are highly branched, and the cells are organized in myofibrils, just like skeletal muscle, and so they have a striated appearance. These cells are connected via intercalated discs that have the gap junctions that allow for communication between the cells, and desmosomes, or anchoring junctions, that will fuse the different cells together. They are also totally dependent on aerobic metabolism for energy, so they need oxygen in order to function. So here we can see another image of the myocardial cells, just kind of reviewing the general structure and how they are branched. Some of the major similarities between skeletal muscle and cardiac muscle is going to be their striated appearance and that they function in very much the same way. However, there is a key difference between skeletal muscle and cardiac muscle from a functional standpoint, and that has largely to do with calcium. This is an excellent chart that shows the similarities and differences between skeletal, smooth, and cardiac muscle. It's great to go through to understand all of this, but I want you to focus on the control row. Control for skeletal muscle happens via calcium and troponin. Remember that calcium will bind to troponin, allowing tropomyosin to move. This will allow the myosin heads to bind to actin and cause contraction. It is also important to remember that skeletal muscle fibers are independent of one another. So not all fibers in an actual skeletal muscle contract, just those that are part of the motor unit that was activated. Let's skip over to cardiac muscle. Cardiac muscle has calcium and troponin as well. Here, the fibers, though, are electrically linked via gap junctions. This means that the fibers aren't independent of one another. It means once one set contracts, they will link and start to contract in a graded function. So this is going to be largely dependent upon the gap junctions and a function of the intercalated discs that hold those cardiac muscle cells together. If we remember back to skeletal muscle, we remember that there was the actions at the neuromuscular junction to where an action potential traveled from the nervous system, activated acetylcholine, and thereby came to the surface of the muscle cell, the sarcolemma, to spread across. From there, if you remember in skeletal muscle, it went to excitation-contraction coupling. In skeletal muscle, excitation-contraction coupling, or EC coupling, is where it's going to travel down to the T-tubules, change the shape of DHP to unlock RYR, which releases calcium. In cardiac muscle cells, it's a little bit different. So here, the action potential is still going to enter from the adjacent cell. It's going to spread across the sarcolemma via those sodium-potassium pumps. Then, as it goes down, it will spread into the T-tubule. So it's the same thing so far. It's going to go down to the T-tubule into that triad region. From here, it's going to activate voltage-gated calcium channels to allow calcium to flow in from the extracellular fluid into the cell. This is a key difference in the actual physiology between cardiac cells and skeletal muscle cells. There is no DHP in a cardiac muscle cell. So calcium is going to enter from the extracellular fluid, and then from here, calcium itself acts as an unlocking agent to the RYR, to the ryanodine receptors. This is going to cause the RYR to open, and the sarcoplasmic reticulum will now release intracellular fluid calcium, which will thereby diffuse down and bind to the troponin, moving the tropomyosin, and from there, we have the actions that are the same as far as contraction relaxation cycling. Remember that to relax, calcium must be removed from the intracellular fluid in two ways. This is the same ways that it would be removed in smooth muscle tissue as well. It's going to have to use the cardiac sarcoplasmic reticulum calcium ATPase pump to pump calcium back into, as you can see here in step 8, the actual sarcoplasmic reticulum. This, of course, uses ATP. Additionally, there's a sodium calcium antiporter, the NCX, that is actually going to pump extracellular fluid calcium out of the cell. This is 9 and 10. That's going to pump calcium out of the cell in exchange for sodium. These are going to be the two major differences from a structural standpoint as far as how the cell returns back to normal after it's going to initiate contraction. And these are going to be the same events that would occur in smooth muscle. These events do not occur in skeletal muscle. Now that we've finished some of the physiology, we're going to move into some of the electrical activity of the cardiomyocyte. Here we actually have the action potential of a cardiac contractile cell. This is going to differ from the actual cardiomyocyte that makes up the pacemaker and the nodal fibers that we'll talk about next. But the actual cardiac contractile cell goes through the following events. First, it's going to start at phase 4, which is resting potential. We start and end at phase 4, so it's not to be confusing, but this is simply going to be the resting membrane potential of negative 90 millivolts. We then go through phase 0. Here, phase 0, sodium channels will open. The voltage-gated sodium channels will open and the cell depolarizes. We then get up to the peak, which is phase 1. Here is initial repolarization. The voltage-gated sodium channels will close and fast potassium channels will open. This allows for an initial repolarization, but it's going to stop short as it gets to phase 2. This is the plateau phase. Here we have voltage-gated calcium channels that are open and the potassium permeability decreases. This is because of the interaction with calcium. So potassium is going to slow down. This is a key phase in this plateau phase. It serves two purposes. First, it brings calcium into the cell for contraction and it prolongs the action potential in this way, therefore the refractory period. This allows the heart to fill with blood. So that's why we have this plateau phase, is to prolong the action potential and allow the heart to fill with blood. Step 3, or phase 3, is rapid repolarization. Here, the voltage-gated calcium channels will close and slow voltage-gated potassium channels will open. This is similar to a nerve in that it will allow the cell to actually go back down to resting membrane potential. Cardiomyocytes do not hyperpolarize. They instead return back to that resting membrane potential because they're already very close to the equilibrium potential for potassium. So here we can actually see a cardiac muscle fiber, how it contracts versus how the electrical action potential works. And this is going to explain that plateau phase to you. So the red line is representative of the electrical action. Here we can see, of course, the resting membrane potential, that depolarization, initial repolarization, and notice the plateau phase. That plateau phase is starting to allow blood to actually fill the heart. So it's going to be filling the chambers during this time and building tension. And from there, the heart will reach peak contraction towards the end of the actual plateau phase when the cell repolarizes. And then it will relax following the repolarization phase. So we've talked about the cardiomyocytes. We're now going to talk a little bit more about the electrical conduction system and then revisit the action potential, this time specifically of the pacemaker cells. The pacemaker itself is the sinoatrial node, or the SA node. This is going to sit up in the right atrium, and it's modified cardiomyocyte cells. This is going to set the pace of the heartbeat at about 70 beats per minute. This, of course, can be quite variable. If you're in better shape, more cardiovascularly fit, you'll typically have a lower resting heart rate. If you are a little bit more out of shape, sometimes it's genetic or you're stressed out perpetually, you are going to have a higher resting heart rate. But the SA node on average sets it at about 70 beats per minute. You then have the atrioventricular node, or the AV node. This is going to actually be where the atria meets the ventricles. This has an actual pacemaker function to set the resting heart rate at about 50 beats per minute. And attached to the atrioventricular node are Purkinje fibers. These are going to they can actually act as pacemakers, setting the heart at 25 to 40 beats per minute under some conditions if the SA node is damaged. Now, the atrioventricular node is actually going to route the direction of electrical signals from the SA node towards the actual apex of the heart. It's going to send electrical signals down to the apex of the heart and then up towards the base. The AV node has a delay in it that is accomplished by slower conductional signals through the nodal cells. This is going to allow the ventricles to fill with blood. This is a simple diagram to show you the difference between pacemaker cells and cardiomyocyte or contractile cells. So the pacemaker cells are autorhythmic cells, and they generate the electrical current. We'll cover their action potential in the next slide. The cardiomyocyte or contractile cells, we've already covered that action potential. They receive the electrical signal from the SA node and carry it throughout the rest of the actual heart. This is going to be the action potential of the pacemaker cell, and this is responsible for cardiac autorhythmicity. It has an unstable membrane potential. This is because it starts at -60 millivolts and constantly drifts towards threshold. Take a look at that bottom. As it's at that resting membrane potential, it's not stagnant. It immediately starts drifting up towards threshold. Now, why does this happen? The slow drift towards threshold starts because there are unique sodium channels that are open at -60 millivolts. As membrane depolarizes, so as it keeps depolarizing, voltage-gated calcium channels will begin to open, and at threshold, it stops, but a lot of calcium channels open, and it depolarizes fully. So we're at resting membrane potential. From there, it's going to drift towards threshold because of the sodium channels that are already open. It's like these specialized sodium leak channels. And it's going to keep drifting towards threshold, and once it hits threshold, it has a rapid influx of calcium that opens. And at peak, calcium channels will then close, and potassium channels will open, allowing the cell to return back down to -60 millivolts. This is when the the potassium channels will close off. But notice it does not hyperpolarize. It simply returns back to normal, and this will have the sodium channels open back up again, allowing for that drift to threshold. So I'll take you through it one more time. We're at resting membrane potential, where a pacemaker potential has a slow depolarization towards threshold. This is because of the sodium leak channels. We have sodium coming in. We reach threshold, -50 millivolts. Calcium channels open up and allow for rapid depolarization, where we hit peak action potential. From there, calcium channels close, potassium channels open, and this allows a repolarization phase as it comes back down to resting membrane potential, and then the whole process starts all over again. Again, this is the pacemaker cell. So your SA node, the AV node has a very similar one to this, but this is going to be the SA node primarily. As you can see from some of the autorhythmic cells, the heart functions in a hyper-coordinated manner in order to pump blood. That means that the individual cells can't contract independently from others. They have to contract in a coordinated manner, so it's a sequential set of events. This also means that the rhythmic contraction of the heart must be controlled and coordinated from a central point. The rhythmicity of the heart originates at the SA node, seen in step 1. And because every cell in the heart is connected via gap junctions, when the SA node fires, the rest of the heart will depolarize. It's like throwing that rock in that pond of water. So the SA node is going to depolarize here. Electrical signals will move from cell to cell via the gap junction and spread through the actual internodal pathways towards the AV node. Once it reaches the AV node, electrical conduction from the atria to the ventricles is blocked except at the AV node. Here, conduction slows through the AV node. We call this AV delay. This allows time for the atria to contract and fill the ventricles. So the filling of the ventricles puts more blood in it and also increases the pressure of the ventricles. From here, depolarization will continue down the septoconduction system. So it's going to go down the AV bundle, into the bundle branches, and then into the Purkinje fibers. As it moves through the AV bundle, also known as the bundle of His, it's going to pass into the Purkinje fibers. So a wave of depolarization is going to travel from the apex, the point of the heart, up towards the base of the heart, which is where you can actually see the valves are. So the very peak of the ventricles. So it's going to spread from apex to base. This is important that it goes from apex to base because it's going to initiate contraction then from apex to base because it's pumping blood up into the actual vessels. It's like wringing out in this spiral muscle arrangement, like you're wringing out of towel. So it's going to be squeezing the heart from apex towards base, pushing blood up into the vessels, and then as it repolarizes, it will repolarize in the opposite direction, from base to apex. So we can see it's traveling up the heart. It's then going to travel down the heart as it repolarizes. This is going to become very important when we look at some of the other electrical activity of the heart, specifically the EKG or electrocardiogram, which we will talk about in the next lecture.